A biofilm is defined as a microbially derived sessile community embedded or encased in a complex 3D gelatinous matrix of extracellular polymeric substance that is secreted by the inhabiting microbes. It is a thick community of cells with a distinct three-dimensional architecture consisting of pillars, towers and mushroom-shaped structures. Through this structure runs an intricate network of channels that provides access to environmental nutrients even in the deepest of the areas and also aids to transport the waste out of the biofilm. A thorough understanding of the architecture has revealed that the establishment, maintenance and existence of biofilm communities is highly complex, socially organized processes, and that the microorganisms in the biofilm exhibit an altered phenotype with regards to growth, gene expression and protein production. Biofilm formation is a phenomena that occurs in both natural as well as man-made environments. It is now well established that 80% of the world's microbial biomass resides in such biofilm state. Most of it is in the natural ecosystems like epithelic communities in rivers, streams, plants and also in the gut of mammals. Biofilms can also form on living tissues, bones and in teeth. Biofilms are relevant in biomedicine as well as in the industry. In biomedicine, biofilms are relevant in clinical setups where biofilms can be formed on prostheses and biomaterials used as implants, or biofilms can form on tissues and on dental surfaces. Biofilms in biomedicine are also relevant in the field of public health when biofilms are formed on water distribution pipes and in tanks, and biofilms that are formed in food processing units. These biofilms can be detrimental because they can cause epidemics. The other area in which biofilms are relevant is the area of industry, where biofilms that are formed pharmaceutical setups where sterile environments are necessary, biofilms that are formed in food processing units and also biofilms that are formed on ship hulls, a phenomena which is commonly called as biofouling. So why do microorganisms form biofilms? Obviously, biofilm formation must be giving some advantage to the microbial population that is growing on the surface. Natural environments often offer a lot of pressures in the form of predation, nutrient limitation, etc. So microorganisms, when they grow in the biofilm form, definitely have certain advantages to overcome these stresses. One of the major advantage that microorganisms get when they grow in a biofilm form is that they are able to resist the physical forces like the changes in temperature, changes in flow and changes in pH. Similarly, microorganisms, when they are in a biofilm form, are not easy for predators like the protozoa to eat. Microorganisms, when they are in the biofilm form under medical setups, you can imagine that the phagocytes, that is the WBCs that are capable of engulfing pathogens, will not be able to easily engulf a microbial biofilm. Similarly, microorganisms in the biofilm also, because of the 3D gelatinous structure, also are able to protect the lower cells from the diffusion of toxic materials. One of the major advantages here that a microbial biofilm in a clinical setup would have is the seepage of antibiotics in the biofilm. It is now well known that microorganisms in the biofilm are almost 100 to 1000 times more resistant to antibiotics because they do not get exposed to the antibiotics that are used for treatment. Biofilms that are formed on surfaces obviously also give an advantage of allowing the microbial population to remain at a particular site in a particular niche. This makes the nutrients more readily available to the microorganisms in the biofilm and also ensures that these nutrients are constantly replenished. Microbial communities in biofilms allow the microorganisms to communicate easily with one another. This type of cell association benefits the microorganisms in the form of nutrient acquisition as well as in the form of genetic exchange. High densities of the microorganisms also provide a framework for coordinated behavior amongst the organisms in the biofilm.